Now, before moving on to the role of pyruvate in breakdown of foodstuffs and the citric acid cycle and the derivation of more ATPs, let's have a brief review of glycolysis using the actual structures of the known enzymes in the glycolytic pathway. And this is a movie that was done by the Cassiopeia Project. It's available on YouTube. And what I've done is added captions and my own voiceover to this movie. So let's take it's about an 8-minute movie, and then we'll move on to the pyruvate, the oxidation of pyruvate, the Krebs cycle, and then electron transport and chemiosmosis. So the first enzyme here is hexokinase. And we have glucose here, these little molecules of glucose, and ATP is involved. Hexokinase will phosphorylate a glucose molecule by removing a phosphate group from ATP. So we're investing energy in this first phase of glycolysis. Here's glucose in the active site, and ATP would enter the active site as well. And then we have phosphorylation of glucose. So we end up with ADP and glucose 6-phosphate are the products. So we now have a phosphate group on the 6th carbon of glucose. Here's the next enzyme, phosphoglucose isomerase. And this isomerase is going to change the shape of this molecule. It isomerizes glucose 6-phosphate into fructose 6-phosphate. It takes 2 molecules of glucose 6-phosphate and makes 2 molecules of fructose 6-phosphate. Here's phosphofructokinase, next step, and it phosphorylates using ATP as a phosphate donor. It phosphorylates fructose phosphate and produces fructose 1,6-biphosphate and ADP. So we've invested 2 ATP molecules so far. We've spent some energy. And now, the fructose 1,6-bisphosphate, we have a primed hydrocarbon ready for breakdown. And aldolase cuts the molecule in the middle, and we have 2 3-carbon molecules produced, each with a phosphate attached. There's multiple subunits in the aldolase protein. And now we have an isomerase, that will transform glycerol, or will transform dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So the one glyceraldehyde 3-phosphate remains. The other one was isomerized to another molecule, glyceraldehyde 3-phosphate. And now each of these can enter the energy yielding steps of glycolysis. Glyceraldehyde 3-phosphate dehydrogenase takes glyceraldehyde 3-phosphate and converts to 1,3-bisphosphoglycerate, putting another phosphate on each of those 3-carbon molecules. Now there are 2 phosphate groups. And finally, we can get some energy out of this. We can take ADPs and we can now phosphorylate them using one of the phosphates from 1,3-bisphosphoglycerate. So we produce an ATP and 3-phosphoglycerate, and we do this twice because we started with 2 molecules of 3-phosphoglycerate. So we have 2 ATPs yielded. Now we're ready to use a phosphoglyceromutase to take 3-phosphoglycerate and move the phosphate group to the middle of the molecule. As we will see here. Now the phosphate's been moved to the center of this 3-carbon compound, and we have 2-phosphoglycerate. And now comes step 9 with enolase. And And enolase will position this phosphate in a thermodynamically unfavorable position, which creates an unstable molecule, phosphoenolpyruvate. And now phosphoenolpyruvate in the last step of glycolysis will be converted to pyruvate, and phosphorylation of ADP to ATP will occur. Finally, we're going to have a net gain in ATP production here because we've invested 2, we've gotten 2 ATPs out, now we get 2 more. 2 more ATP molecules. And phosphoenolpyruvate is converted to pyruvate, two pyruvates, which can then leave glycolysis and either oxidize completely to carbon dioxide and water in aerobic respiration, or in fermentation converted to ethanol, alcohol, or lactic acid in skeletal muscle. And here's a whole review of the process with the intermediates shown. We started out with glucose, and now we have various enzymes. And so it would be helpful, this is a nice review for you to kind of just look at those intermediates and see how many you can identify, and see how many of the enzymes you can identify as well, just to see if you've learned the glycolytic pathway in great detail. Most importantly, if you could identify the ATP producing steps in the energy yielding phase, that would be very useful. You would find that you understand glycolysis pretty completely then. Finally, we end up with pyruvate at the very end. And now it's on to pyruvate oxidation, the Krebs cycle, and electron transport.